Hey guys, welcome back to another video tutorial from my channel Aditya Byte. So in this video tutorial we will discuss on cellular respiration. Okay, we will discuss the overall concept on cellular respiration, also about the detailed process of glycolysis in this video. So let's start. So as we all know what the cellular respiration means. So cellular respiration is basically the process of breakdown of complex organic molecule into simpler one to form metabolic energy. So guys, this is the basic concept here guys. Okay, so let's move on now. Now let's know the overall view of cellular respiration. So actually what happens is glucose is preferred as main substrate here. The glucose is being converted into pyruvate by a process called glycolysis. Okay, and after to that, for the further breakdown of pyruvate, it depends on the basis of whether the oxygen is present or not present. As you can see here, if not present, then it goes for a fermentation process for the formation of ethanol or lactic acid. Okay, and if present, it goes for a different phases like oxidative decarboxylation, Krebs cycle, electron transport chain, and up to 36 ATPs are generally generated. Okay, and so these phases are also called aerobic phase, aerobic respiration phase, and this phase is called anaerobic respiration phase. This is the basic concept here guys. Okay, now let's move on. So today in this video lecture we will discuss on the detailed process of glycolysis. As you can see, glycolysis is made of two words, glyco and lysis. Glyco means simply glucose, okay, and lysis means breakdown of that glucose. These were the basic concept, and glycolysis mainly takes place in the cytoplasm. Okay, and this is the first step of respiration here. Now let's see the overall concept on glycolysis. Now glycolysis, as we have already discussed that glycolysis, glykis means sweet, and lysis means splitting. Okay, now let's see the process in cell metabolism by which carbohydrates and sugars, especially glucose, are break down into ATP, pyruvic acid, and two high energy electron carrying molecules of NADH. And this NADH further goes in the ETC cycle for the release of energy in the form of ATP. This is the basic concept here guys, and this is the structure of glucose here as you can clearly see. Now let's move on. So this is the arrow diagram. This arrow diagram indicates the whole process of glycolysis here. Here what happens is glucose breakdowns and forms two molecules of pyruvate, which is the 3 carbon compound. Okay, and also with it 2 ATPs and 2 NADHs are also released. Okay, so this was the basic concept for glycolysis here guys. Now let's move on. Now let's discuss the overall steps in glycolysis. In glycolysis, 10 steps occur, okay, and out of which 7 steps are reversible steps and 3 steps are irreversible steps. This was the basic concept here guys. Now let's see what happens is here glucose converts into glucose 6-phosphate with the release of with the use of energy in the form of ATP. As you can see, ATP is being converted into ADP, and this is accompanied by an enzyme hexokinase glucokinase. Okay, and after to that glucose 6-phosphate converts into fructose 6-phosphate with the help of an enzyme phosphohexose isomerase. This is nothing but a rearrangement reaction in which glucose 6-phosphate is being converted into fructose 6-phosphate, which are also the isomers here, and this is irreversible reaction. This is a reversible reaction actually, you can see. Okay, And then after that, what happens is fructose 6-phosphate, it converts into fructose 1,6-bisphosphate. With the help of energy, ATP being converted into ADP, and enzyme acting here is phosphofructokinase. Okay, this was all about. And then after that, what happens is the splitting occurs. Here you can see fructose 1,6-bisphosphate is being splitted into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. These are actually the isomers which are linked together by triose phosphate isomers and are reversible reactions here. Now let's move on and see. Now let's see glyceraldehyde 3-phosphate. What happens actually after to that? Glyceraldehyde 3-phosphate converts into 1,3-bisphosphoglycerate. Okay, with the help of an enzyme glyceraldehyde 3-phosphate, and here an inorganic phosphate molecule operates. Okay, and NAD plus inorganic phosphate converts into NADH plus H plus. Okay, this was all about. And after to that, what happens is 1,3-phosphoglycerate converts into 3-phosphoglycerate. Here you can see one phosphate group is being removed and is converted into 3-phosphoglycerate, and that phosphate group is utilized in the formation of ATP, as you can clearly see here, guys. And then after to that, 3-phosphoglycerate is being converted into 2-phosphoglycerate with the help of an enzyme phosphoglycerate mutase. Okay, and then after to that, this 2-phosphoglycerate converts into phosphoenol pyruvate with the help of enzyme enolase, and then this phosphoenol pyruvate converts into finally the pyruvate, our product, with the release of ATP molecule here, and with the help of an enzyme pyruvate kinase. Okay, and guys, this phase, which is also called the ATP generation phase, occurs 2 times, as dihydroxyacetone phosphate also converts into glyceraldehyde 3-phosphate after. So this was all about, guys. Okay, and this phase, as we have discussed, this phase is called the investment phase. Okay, and this is the splitting phase. Okay, and this phase is called the ATP generation phase, as we have already discussed. This was all about, guys. Now let's see the steps one by one for more clearance. Okay, now let's start. So 10 steps in glycolysis. So first step is, what happens is this glucose is being converted into glucose 6-phosphate. Okay, as you can see, the phosphorylation occurs here. Here the one phosphate group is being attached in the glucose here. Okay, and this is the irreversible reaction, and ATP is being invested here, as you can clearly see. Glucose receives a phosphate group from ATP to form glucose 6-phosphate. Process phosphorylation, enzyme hexokinase. Okay, now let's see other step. Now second step is glucose 6-phosphate converts into fructose 6-phosphate with the help of an enzyme phosphohexose isomerase. Okay, which is a reversible reaction also. Glucose 6-phosphate is isomerized to fructose 6-phosphate by an isomerase. This is reversible reaction. Yeah, this is actually the rearrangement reaction actually. Now let's move on. Now third step is fructose 6-phosphate is converted into fructose 1,6-phosphate. Here one ATP is invested here, and the enzyme acting here is phosphofructokinase, and one additional phosphate is actually added here, as you can clearly see. Yeah, fructose 6-phosphate is phosphorylated to fructose 1,6-biphosphate by an enzyme phosphofructokinase or PFK. Energy for this reaction is derived from ATP, and PFK is important key enzyme of this pathway. This is irreversible reaction, as we have already discussed. Let's move on now. Now the another step is in which fructose 1,6-bisphosphate is converted or is being splitted into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate with the help of an enzyme adolase. Fructose 1,6-bis phosphate is splits into two molecules as I already discussed, glycerol-3 phosphate and dihydroxyacetone phosphate. Enzyme is aldolase and this is also reversible reaction. Let's move on now. Now let's see the splitting in detail. Dihydroxyacetone phosphate and glycerol-3 phosphate as it has been splitted upon. Dihydroxyacetone phosphate is isomerized to glycerol-3 phosphate by the enzyme phosphotriose isomerase. In this reaction, two molecules of glycerol-3 phosphate are formed as we have already discussed, and glycerol-3 phosphate can also be synthesized from glycerol by phosphorylation. This is reversible reaction. Okay, let's move on now. Now in the sixth step you can see glycerol-3 phosphate is being converted into 1,3-bis phosphoglycerate. In this, the additional phosphate group comes from the inorganic phosphoric acid, okay, as 3PO4, and this NAD plus is being converted into NADH, and this NADH further goes in the mitochondria for the processing in ETC cycle and the release of 6 ATP from 2 NADH. This was the basic concept here, guys. Now let's see, glycerol-3 phosphate gets dehydrogenated and phosphorylated to 1,3-bis phosphoglycerate by G3PDH. Here you can see, and reducing equivalents are carried by NAD plus. This is reversible reaction. This was the overall concept. Now let's move on. Now another step is the seventh step. The 1,3-bis phosphoglycerate is being converted into 3-phosphoglycerate, and here ATP is released. Okay, actually what happens is one phosphate group is being released, and that is helped in the utilized in the formation of ATP here. This the energy of 1,3-bis phosphoglycerate is used for the synthesis of ATP in this reaction. We have already discussed. This is example of substrate level phosphorylation. Yeah, here energy is trapped directly from substrate without involvement of ETC. Here ATP is forming directly without going in ETC cycle. Okay, guys, this is reversible reaction, and arsenate inhibit this reaction. This was all about. Let's move on now. Now in the eighth step, what happens is 3-phosphoglycerate is being converted into 2-phosphoglycerate. And this is nothing but the shifting of phosphate group. Here you can see the phosphate group is being shifted to here now, from third position to second position. This was all about, and this is a readily reversible reaction, guys. Okay, let's move on now. Now the ninth step is 2-phosphoglycerate is being converted into phosphoenol pyruvate. Okay, with the help of an enzyme enolase here. Here 2-phosphoglycerate is converted into phosphoenol pyruvate by an enzyme enolase by removing one water molecule. Okay, this is a reversible reaction, and enolase requires magnesium here, important term, for their reaction, and fluoride irreversibly inhibit enolase, thereby stops the whole glycolysis. Okay, therefore fluoride is added to blood during estimation of blood sugar. This was the concept, guys. Let's move on now. Now the last step, the 10th step, in which what happens is phosphoenol pyruvate converts into pyruvate with the release of an ATP, the energy here. Okay, and this phosphoenol pyruvate converts into pyruvate by the enzyme pyruvate kinase. Okay, here also energy of PP is trapped into ATP as we have already discussed. This is the example of substrate level phosphorylation again. Okay, and pyruvate kinase is a key glycolytic enzyme. This reaction is irreversible as we have already discussed, that in the whole cycle three steps are usually irreversible steps, and they were the step 1, step 4, and the last step 10. This was all about, guys. This was the basic concept for the glycolysis, and after the formation of this pyruvate, the further breakdown occurs in the basis that whether the oxygen is present or not present in the mitochondria, as we have already discussed. Now let's move on. Now let's see the energetic of glycolysis. In this process, in this step, what happens is the net gain of ATP is calculated here. The net gain of ATP in glycolysis is usually 2 here, Actually what happens is two ATPs are already invested in the investment phase, and after to that in the ATP generation phase 4 ATPs are generated. So net ATP formed is equals to 2 ATP. Okay, and also 6 ATPs are formed from the NADH by ETC cycle, as we have already discussed. Yeah, so actually the net gain of ATP in the glycolysis is only 2, and the total production of ATP in the glycolysis is actually 8. In glycolysis is actually 8. Okay, that is 6 from NADH and 2 from the ATP directly formed. This was all about, guys. Let's move on now. Now let's see the steps in overall. In the last, let's recall the steps here. Here as you can see, glucose is being converted into glucose 6 phosphate, then fructose 6 phosphate, then fructose 1,6 bis phosphate, then the splitting is occurring, then the glyceraldehyde 3 phosphate being converted into 1,3 bis phosphoglycerate, then 3 phosphoglycerate, then 2 phosphoglycerate, and phosphoenol pyruvate PEP, and then pyruvate. And this pyruvate, the further breakdown of pyruvate occurs in the basis that whether the oxygen is present or not in the Krebs cycle, in the ETC cycle, which takes place in the mitochondria. This was all about, guys. And here in 10 steps, 7 are reversible steps and 3 are irreversible steps. Okay, so this was all about, guys. This was all about the glycolysis process. So I hope the glycolysis process is being clear and being simple to you now, guys. So if you like the video and if you found the video effective, then please hit the like button, share and subscribe the channel for more such upcoming videos. So for today, this much only. Thank you. Bye bye.